So in the evolutionary arms race between bacteria and antibiotics, antibiotic resistance has become more and more prevalent, particularly since we're seeing the misuse and overuse of antibiotics. Alternative and potentially new treatments have become more and more important to discuss. So in fact, the World Health Organization predicts that by 2050, the drug resistance crisis will cause the global economy more than $100 trillion US and be the cause of more than 10 million deaths yearly. Now, how did this come to be? Well, the 21st century has actually seen a sharp decline in the development of new antibiotic classes, leaving us dependent on the previously established ones. According to the Centers for Disease Control and Prevention, about 1/3 of all antibiotic use in people is actually not needed nor appropriate. Now, due to this overprescription and misuse of antibiotics, we are currently seeing high morbidity and mortality rates among antibiotic-resistant bacteria, being 2.8 million infections and 35,000 deaths yearly just in the US alone. Currently, according to the Centers for Disease Control and Prevention, the most prescribed antibiotic classes in the US are penicillins, macrolides, cephalosporins, fluoroquinolones, and beta-lactams with increased activity. These antibiotic classes can then be classified into three more broad classes known as beta-lactams, macrolides, and fluoroquinolones. So now that we rely mostly on these antibiotics, which have been developed several decades ago, this signifies that there are actually very high selective pressures on susceptible bacteria to gain resistance mechanisms and genes currently. So in order to better understand and really evaluate our potential solutions to the antibiotic resistance crisis, we must first understand the mode of action of these antibiotic classes before then diving into the complementary resistance mechanisms developed by bacteria against these classes. Only then can we actually start to better comprehend and evaluate alternative non-antibiotic therapies while considering their future implications. So we'll be looking at the three main classes, as I mentioned. So beta-lactams are they consist of three subclasses. So penicillins, cephalosporins, and carbapenems. And these classes all have in common a highly reactive beta-lactam ring. It's a four-membered ring, which is responsible for the inhibition of the synthesis of the peptidoglycan layer. So as shown in this figure, the peptidoglycan layer is primarily composed of a polypeptide and a polysaccharide chain. So these beta-lactam molecules, specifically the rings, are responsible to react with and bind with an enzyme known as transpeptidase. So this enzyme transpeptidase is responsible for the facilitation and catalyzation of these bonds in the peptidoglycan layer between the polysaccharides and the polypeptides. So by essentially rendering this the active site of this transpeptidase enzyme inactive, these beta-lactam molecules inhibit the complete synthesis of the peptidoglycan layer, acting as suicide inhibitors for bacteria. On the other hand, macrolides actually hinder protein synthesis during the process of translation. So shown in this figure is the bacterial ribosome, which is composed of two subunits, the large 50S subunit on the top, and then the small 30S subunit on the bottom. So how macrolides work is that they cause a suboptimal orientation of specific mRNA sequences by narrowing the nascent peptide exit tunnel, also known as NPET, which is found on the large 50S subunit. So in this figure, we can see that the macrolide molecule is actually narrowing that tunnel, which causes specific arrests during the translation process, essentially hindering protein synthesis. Finally, fluoroquinolones are responsible for targeting two essential bacterial enzymes known as the topoisomerase 2 and topoisomerase 4. These enzymes are responsible for ensuring the proper transcription of all bacterial genome during the process known as binary fission. So by bonding to the active site of these two enzymes, fluoroquinolones actually ensure that the bacterium cannot replicate itself, therefore preventing and acting as a suicide inhibitor. Okay, so according to Kieran and other researchers, resistance mechanisms can actually be grouped into three main classes or types. So first being enzymatic, biochemical, and then genetic. Oh, okay, there we go. So beta-lactamase are part of the enzymatic mechanisms, and these are these enzymes found in antibiotic resistance bacteria. So they're responsible for the inactivation of the four-membered beta-lactam ring. And they do so by hydrolyzing the ring, causing it to open. So as we can see in this figure, this is an example of a serine penicillin molecule, and the beta-lactamase is actually causing the four-membered beta-lactam ring to open. And since this beta-lactam ring is the segment of the molecule that causes it to react and actually be effective, by inhibiting it and actually inactivating this ring, causing it to open, it actually renders the entire molecule ineffective. Furthermore, the next enzymatic mechanism is group transfer. So group transfer is a mechanism done by the largest, most diverse enzyme family called transferase. And these enzyme families, this one is responsible for the inactivation of numerous drugs by impairing its ability to bind to its designated target site. So an example of this group transfer mechanism would be the phosphorylation. So phosphorylation occurs when these enzymes, the transferase, actually add one or two phosphate groups to the drug molecule, rendering it completely ineffective. Okay, so there are three main biochemical mechanisms that we'll be going over today. So first being ribosomal methylation, then penicillin binding protein structure modification, and drug efflux. So ribosomal methylation is prevalent in gram-positive bacteria. And this is caused by these genes known as erythromycin ribosome methylase, or ERM genes. And these genes are actually going to be transcribed and translated to produce an enzyme called methylase, which is responsible for altering the adenine residue in the large 50S subunit of the bacterial ribosome, which then overall decreases the subunit's affinity for antibiotics. Furthermore, penicillin binding proteins are actually the main target of the beta-lactam class. As we saw, the beta-lactam class is a very, very broad class of antibiotics. So by altering the penicillin binding proteins, they actually render most of these antibiotics ineffective. So there are these certain genes that can be either horizontally transferred or just kind of gained as foreign genome. And these encode the penicillin binding proteins for a decreased affinity for the beta-lactam ring, and therefore do not react with it. Lastly, drug efflux. So these are efflux pumps that are proteins that are found in the bacterial cell wall or in biofilm complexes, with the latter being responsible for more than 65% of all bacterial infections. So these proteins, these pumps, essentially help the bacteria manage its internal environment by letting it expel harmful solutes, such as the antibiotic molecules. So as we can see in this figure, these pumps are actually classified into 6 different classes, but then they have many different types and 33 subtypes. So there's a lot of them, but this figure kind of shows some most commonly seen drug efflux pumps. Lastly, the only genetic mechanism that we'll be going over today is plasmid transfer. So this mechanism is adopted by 50% of all bacteria in nature. The horizontal transfer of plasmids, which is a small circular segment of DNA, as we can see in the photo, actually is one of the main mechanisms for the transfer of resistance genes. So since these little plasmids can carry multiple resistance genes at a time, they actually can lead to MDR, which is multi-drug resistance, which renders multiple different antibiotics ineffective at the same time, which is very dangerous. So this figure shows one of the multiple ways that this could happen, that plasmids could be transferred. And this specifically shows the process of conjugation, where which consists of cell-to-cell contact between the donor and the recipient bacteria cell. So the end result of the conjugation is that both cells end up with an identical plasmid. Now for alternative therapies. While there are numerous different alternative non-antibiotic therapies being developed and tested as we speak, unfortunately, for the sake of time, I'll only be presenting two main ones, namely bacteriophage therapy. I think the Bluetooth connected. Okay. Oh, is it Siri? No. Okay. Sorry about that. So bacteriophage therapy, also known as PT, is very, very effective as lysing bacteria cells. Actually, in fact, in high dosages, it can terminate a bacterial infection completely. So when using bacteriophage therapy, it is generally advised to use lytic phages instead of lysogenic phages, since they're generally more efficient at lysing bacteria cells. While this is very beneficial, it has a lot of limitations. So most bacteriophage therapy actually consists of phage cocktails, meaning that there are several different types of phages being used to being introduced into the patients, meaning that there is a broad host range. However, there are also equally bacteriophage therapies where only one single type of phage is being used, allowing for a more personalized approach. Regardless, its host range is very, very narrow compared to the host range of most antibiotics, putting it at a disadvantage. Furthermore, phages can also be a way of transferring resistance mechanisms, which will then add on to the antibiotic resistance crisis, which in turn is not beneficial at all. Furthermore, antibacterial vaccines are classified into 4 different types. So subunit vaccines, toxoids, live attenuated, and inactivated vaccines. So antibacterial vaccines actually offer several advantages over antibiotics, 2 of them in particular being the timing of treatment and the multiplicity of targets. So antibacterial vaccines are prophylactic, whereas most antibiotics are used in a therapeutic manner, meaning that their timing of treatment differs. So whereas antibiotics may be used when the patient is showing some symptoms, suggesting that there are already billions of pathogenic bacteria present in the host system, antibacterial vaccines are used in a preventative measure, meaning that the host organism has already forged an immune response long before and during the early stages of the infection. Furthermore, most antibiotics actually consist of one single target, whereas antibacterial vaccines actually target multiple antibody-inducing targets. Some limitations of the antibacterial vaccines is that they may be subject to a poor immunogenic response from the host, so it really differs from subject to subject. And furthermore, they have a very limited host range. Since they are targeted specifically towards one bacterial strain, they do not cover multiple bacterial infections at the same time. So they are at a disadvantage in that way. So concluding remarks, evidently the antibiotic resistance crisis is very urgent and very threatening to human health and the very foundations of medicine as we know today. As a result, it is very crucial that we not only explore new alternative therapies, but definitely evaluate them while considering their current and future implications. Nonetheless, the road to finding effective and practical potential solutions to the antibiotic resistance crisis is a very long one with lots of obstacles along the way. So it is crucial that we do more research, and more research not only on new solutions, but also on better understanding and further understanding the antibiotics mode of action and also resistance mechanisms. Furthermore, future studies can equally help us understand new and emerging mutations and other leading factors that may result in new and emerging resistance mechanisms. Thank you. And I just wanted to conclude this presentation by thanking my mentor, thanking Talaria for everything. I think working with my mentor, Antoine Pelletier, has definitely been the greatest part of this whole experience. He's definitely an amazing, amazing mentor, amazing researcher. And he was there, not only there to guide me, but actually help motivate me too, because sometimes I felt a little bit burnt out since this is a literature review and we were going through lots and lots of academic papers, and I didn't really have that much experience with analyzing and reading papers at such a great quantity. So I think definitely he was always there when I needed him, and he always provided feedback. And for sure Talaria, because this whole experience would have not been possible without the Talaria Research Institute. So thank you, everyone.